As of the 23rd of August 2025, the COVID-19 pandemic has claimed over 7 million lives. The rapid global spread of the SARS-CoV-2 virus, along with slow government responses, made curbing the pandemic difficult in its initial stage. One major hurdle is the availability of high-quality 3D structures of viral proteins. Traditional methods, such as NMR or X-ray crystallography, can take months. Any vaccine development strategy would need 3D structures to understand how the virus functions and interacts with host proteins. The COVID-19 pandemic saw the rise of 3 major variants of concern after the original Wuhan strain: Alpha, Delta, and Omicron. This meant that scientists were in a race against time to obtain high-quality experimental structures of SARS-CoV-2 proteins when a new VOC emerged. Deep learning technologies, such as AlphaFold, can now keep pace with the emergence of new mutations and VOCs, as they can now be modelled with very high accuracy in a fraction of the time. This reduces the reliance on experimental structure prediction. We can now map out the structural impacts of mutations before we obtain experimental structures, and use that knowledge in vaccine development strategies. However, VOCs are not the complete picture, and the viral population always contains a mixture of groups of mutations that coexist with the current dominant VOC. Mutations that were part of VOC Omicron existed before VOC Alpha appeared. Groups of mutations that showed similar accumulation patterns are grouped into a haplotype, which can be studied further. During the pandemic, the 3D structures that were released were usually of well-established variants. Those of haplotypes were not available, which is why we used AlphaFold to model 13 haplotypes that impacted the SARS-CoV-2 spike protein. A thorough comparison of each haplotype and VOC revealed an interesting pattern of recruitment of substructures. Some haplotypes were recruited in all 3 major VOCs, whereas others were not. The haplotypes existed long before the VOCs, and they usually kept increasing in frequency until they were recruited into the dominant VOC. This recruitment process can cause unexpected changes to the spike protein. VOCs Alpha and Delta seem to behave antagonistically by suppressing the structural changes brought about by their individual haplotypes, whereas Omicron seems to behave synergistically by amplifying the effects of its individual haplotypes. Another interesting finding was that most of the structural changes were found to be in loops or regions of intrinsic disorder. These regions play an important part in viral function, such as the furin cleavage site, or are part of epitopes. We also noted that the N-terminal domain hosted 8 out of 21 reported structural changes, with many coinciding with the NTD supersite that is part of an epitope with neutralizing capabilities. The haplotypes impacting NTD structure the most were noted to behave seasonally when looking at their accumulation plots over time. Vaccine development strategies can benefit from using this approach of analysing haplotype structures, as it can shed light on how future variants may behave.